Hello and welcome to another episode. So we'll be discussing about the MCQs that I had put up in my Instagram page. So if you're not following it yet, you can check out this address over here. And so we'll try to solve the MCQs that were given during the first week. Gastric inhibitory peptide is released by which of the following cells in the GIT? So the answer for this is going to be your K cells. Okay. So let's take a look at all the cells that release different hormones. So G cells release gastrin. So G for gastrin. D cells somatostatin. So this both are there in the stomach. The S cells release secretin, and I cells release cholecystokinin. M cells release motilin. The K cells release gastric inhibitory peptide. The N cells neurotensin. Paneth cells guanylin, and the ends of vagal neurotransmitter release is going to be your gastrin releasing peptide. So the answer for the question was K cells, which release gastric inhibitory peptide. Juxtaglomerular apparatus contains all except. So we have macula densa, lassus cells, juxtaglomerular cell. Glomerular mesangial cell is not part of this. So that is the answer. So now let's take a look at trying to understand what is making up your juxtaglomerular apparatus. Okay, so please keep in mind that juxtaglomerular cell and juxtaglomerular apparatus are two separate things. So you can see over here the efferent arteriole, and in the efferent arteriole we have the juxtaglomerular cells which are there. And you can see here this is the glomerulus, and from there it goes on to form the PCT, the descending limb, ascending limb, this is a loop, and distal convoluted tubule. So when the thick ascending limb becomes the distal tubule, around that area is where it comes opposite your whole glomerulus, and that is where specialized cells called the macula densa are present in the distal tubule. Okay, so one is your juxtaglomerular cell, the other is a macula densa, and between them, between the efferent arteriole and the macula densa, you got mesangial cells called the lassus cells. Keep in mind that even here inside the Bowman's capsule, uh, between these capillaries, you can see a type of mesangial cells. So these are your glomerular mesangial cells. So they do not make up a part of your juxtaglomerular apparatus. Okay. So again, juxtaglomerular apparatus basically includes your lassus cells, the juxtaglomerular cells which are over here, and your macula densa. Oxygen hemoglobin dissociation curve is sigmoid because. So here there are options talking about binding of one oxygen molecule, decreasing the affinity of binding more, binding of one oxygen, increasing the affinity, and oxygen affinity of hemoglobin changing with the pH, and binding of O2 to hemoglobin affecting CO2. So the answer for this is going to be this. Basically, when one molecule of oxygen binds, it kind of increases the affinity of oxygen and hemoglobin. So you can see over here the sigmoid shape, and in the sigmoid shape, what you can notice is the two ends are where they're kind of slower. Okay, when it begins, and at the top. In between, the rate is kind of higher. The slope over here will be much higher. Okay, so the rate is much higher here. So the reason for the sigmoid shape is because of something called cooperative binding. So you can see over here the hemoglobin molecule. Okay, this is supposed to be a hemoglobin. Just for representation, we have the alpha and the beta globins over here. Okay, so now one oxygen molecule comes in, associates itself with the hemoglobin, the heme part of it, and that will kind of make it change its state. Kind of slowly it goes from a tense state, it goes into a more relaxed state, and again that increases the affinity. So after this, again greater affinity, and again when more binds, greater affinity. Okay, so that is why initially it's lesser. Once enough has bound to it, it basically increases the affinity, resulting in this shape. And towards the end, after the third one, so at the third one it reaches a maximal state, after which not much difference comes by the time fourth binds. So that is why again it's slow down. Okay, so this is what is called cooperative binding. And basically what happens here is the tense state becomes your relaxed state, and that opens up your oxygen to come inside and bind to the heme part. Which of the following is not related to caveolin vesicular transport? So cavin, dynamin, caveolin are all related to caveolar mediated vesicular transport, and triskelion is for your clathrin mediated transport. So you can see over here the lipid raft, that part of the membrane which is rich in cholesterol, sphingolipid and all that. Okay, and it is in this region that an integral protein called the caveolin is attached. And apart from that, there are peripheral protein called the cavin which are also present, and these take part in your caveolar mediated. And you can see over here the dynamin. Okay, so that helps it pinch off. Now these are the things that are involved in caveolar mediated vesicular transport. This triskelion structure is what is there in clathrin mediated vesicular transport. So this triskelion structure is what is essential to make the clathrin cages. False statement among the following. So baroreceptors are more effective in the range of 50 to 200, so that is true. In response to increased BP, maximum number of impulses at 180 mm mercury, so that is also true. Carotid baroreceptors not stimulated below 50 mm mercury, that is also true. The rate of rise of signal for rise in BP is maximum at 180, so this is false because the answer is going to be 100. Okay, so we'll take a look at the graph to understand this. So over here, the number of impulses is what is shown here from the baroreceptor, and this is your BP. So as the BP increases, the number of impulses also increase. But again, it's showing a bit of a sigmoid pattern. So what you have to understand is below 50 to 60 range. Okay, so that point where it is around 50 to 60, below that you can see that there are no impulses coming. Okay, so at that value of low BP, below 50 to 60, there is no impulses. After that, the impulses keep increasing, and you can see that the rate is going to be increasing till at one point around 180 millimeter mercury, it kind of forms a plateau. Okay, now this part over here, that is where the rate of change per unit change in pressure is going to be maximum. So in the whole sigmoid pattern, this is the point where the slope is the maximum. So the rate there is maximum. So for a unit change in pressure, the maximum without lag, the most effective point is going to be at this. Okay, and what you have to remember is your mean arterial BP comes to around your 100 millimeter mercury, just like this. So that is when it is most effective. And it has a range between your 50 to 200 millimeter mercury. Slow rhythmic movement due to lesions in putamen. So putamen lesion, and it's a slow rhythmic movement. The answer is going to be chorea. So in chorea, what is going to be there is some sort of involuntary, irregular, unpredictable dance-like movements. Okay, so there'll be like unpredictable, involuntary, irregular, and dance-like movements which are there, as you can see over here. When you compare it with athetosis, athetosis is a much slower process. It's more continuous, slow. It's also involuntary, but a rhythm type of movement. Okay, so you can see how a person moves like this continuously, like this. So that sort of a movement is athetosis. Ballismus, on the other hand, is mostly affecting the proximal joints and is high amplitude movement, ballistic sort of movement. Okay, so they're like sudden movements, like the sudden violent jerks. Okay, with high amplitude ballistic movements that affects the proximal joints more. So this most often is going to be because of lesions in the subthalamic nucleus. The other hand, your chorea athetosis has got to do a lot with your striatum pattern. Okay, so it's a little interchangeable actually based on studies. Different things give different things. But for your exam, you can just remember that putamen is chorea. Your lenticular nucleus is going to be your athetosis. Lenticular nucleus also contains your putamen. Okay. And subthalamic nucleus is Ballism. Okay.